May I now invite Professor Michael Young to chair the following session.
Okay, good afternoon. And now we come to the more important part. So again, it's my great pleasure to welcome everybody here and online to join this inaugural AFCR-CityU Innovation Series in Biomedicine. We have a very big feast of intellects this afternoon, and so please allow me to introduce our first speakers, Professor Bruce Johnson. Professor Johnson is currently the Institute Physician in the Dana-Farber Cancer Institute and Professor of Medicine at Harvard Medical School and the Brigham and Women's Hospital. He is a co-leader of the Dana-Farber Harvard Cancer Non-Cancer Program and co-director of the Center for Cancer Genomics at the Dana-Farber Cancer Institute. Professor Johnson received his undergraduate degree from Harvard and his medical training at the University of Minnesota and internship internal medicine at the University of Chicago. Prior to joining Dana-Farber at 1998, he was working at the NCI for several years. He came to Dana-Farber in 1998 and served in the Lower Center for Thoracic Oncology and served in that position until 2017, and then transitioned to be the Chief Clinical Research Officer of the Dana-Farber Cancer Institute until 2021. He is now the Senior Advisor to the President and CEO of the Dana-Farber Cancer Institute. Professor Johnson is also the President of ASCO, American Society for Clinical Oncology, during 2017 to 18, and also contribute a lot to the development of the cancer research network, both in the US and globally. And Dr. Johnson was one of the investors who helped develop the EGFR inhibitor, one of the most famous and most important cancer target therapies that save, I would say, thousands, if not millions of lives over the years. And so without further ado, let's welcome Professor Johnson to give us a lecture on precision medicine applied in non-cancer.
Thank you, Professor Yang and Professor Ba, when you first put it up there. All right, thank you. I'm going to be, one of the things is I both run laboratories and treat people with lung cancer, and I've been able to witness the changes that have taken place over the last 2 decades that have changed not only how we treat people, but also on patients' outcomes, and not only ones with a specific change, but it's also begun to have an impact on how long people live with lung cancer. And what I want to do is go through a few examples of how we've studied the individual characteristics of the patients that informs their treatment and how we perfect that and move forward. The second thing I want to do is give some examples of things that have come along the way that we've been able to discover and license to companies that have given income to the Dana-Farber Cancer Institute and my family. So I want to talk first about the first step toward precision medicine and how long it took, because I want to show you the accelerating pace of discovery moving to actual treatments that have an impact on people's cancers. And that's something called a BCR-ABL. It's a breakpoint cluster rearrangement. ABL is the name of a gene. And the first one that happened was in chronic myelogenous leukemia. And I want to show you how long it took between the discovery of this and when they had medicines, but also importantly about how it's had an impact on the treatment of patients who have that, what was originally a very deadly disease. So it started in 1960, and this is a picture of a chromosome. And chromosomes are numbered from the biggest one to the smallest one. So chromosome 9 is bigger than chromosome 22. And Dr. Knoll and Hungerford in 1960 discovered that this bottom piece of chromosome 9 moved to the part of chromosome 22 and formed a fusion protein called a BCR-ABL translocation. So the initial discovery was seeing this at the chromosomal level. Then it took 22 years before they could end up sequencing this and finding out that this thing is called an Abelson tyrosine kinase. So we're going to talk a bit about tyrosine kinases, which are druggable proteins. And then it took another 14 years before they came up with a compound that could inhibit this activated Abelson kinase, and that drug is called imatinib. And that drug is active against this Abelson, and by that translocation is highly activated. The chromosomes break, rejoin, and the Abelson tyrosine kinase is highly expressed, giving a therapeutic advantage for this. And as I mentioned before, this was discovered back in, this was the medicine that you could use it for was discovered in '96. So the clinical trials started with imatinib, which is a drug that's directed against the Abelson tyrosine kinase. And it was approved in the United States in 2001. So it took 41 years between the discovery of that chromosomal rearrangement and an approval of a drug. So not many of us have very many of those in our careers. You know, most of our careers span 40 years, so you can't do that very often. Now, the one thing it did do is it had a dramatic impact on the outcome. So as I mentioned before, imatinib for this disease was approved in 2001. Before 1975, you know, which is still 15 years after discovery, 6% of people made it to 8 years. Almost everybody died in that time period. And after imatinib, after 2001, 87% of people were alive at 8 years. So this discovery that took 40 years ended up transforming the outcome of these patients. And so this is the first targeted oncogene, and it gave us an example of what we might do if we could discover other genomic changes where you could make different targets. Now, one of the things I'm going to be doing here is not only focusing on these drugs that can affect these activated cancer genes, but also how you can take a look at overcoming cancer resistance. One of the things that has had an impact on our efforts in the United States that hopefully will be paralleled here in Hong Kong is that they announced what we call a cancer moonshot, and the U.S. ended up putting about $1.5 billion extra into cancer research, and they generated 10 priorities, and one of these 10 was to develop ways to overcome cancer's resistance. So I'm going to show you a couple of examples of studying how well the drug works, finding out why it quits working, and then develop drugs that work against it, which, as I said before, was one of the priorities. This is a picture of President Obama speaking at his State of the Union. That's where our president talks about what he hopes to have happen in the coming year. This is our current president, who was vice president at the time, and he had a son who died of a brain tumor. And he's very interested in cancer research and has spearheaded that effort. For those of you, this is our Speaker of the House. It's one of the, it's the other person that is present and sits behind at the State of the Union address. So the one I'm going to talk about is EGFR mutation. So, you know, in the US and Europe, it's about 10% of patients, but here in China, it makes up, you know, between 40 and 50%, depending on where you are in China, of all the patients who come down with lung cancer. And I'm going to talk about, you know, what we found out 20 years ago and what's happened and what we've been able to commercialize. So I want to, similar to what I showed you before, about 6% of people with chronic myelogenous leukemia being alive at 8 years, I want to show you where we were 20 years ago in treating lung cancer patients. So this is when I first was getting started in lung cancer, and at the time they were trying different combinations of chemotherapy, and it's not terribly important. These are 4 different doublet regimens that at the time were being used for advanced lung cancer. And they studied 1,200 patients, a very large trial, and came out with virtually the identical outcomes. And one of the things I want to point out here is that only 11% of people are alive at 2 years. So, and at 5 years, it was similar to what we saw with imatinib, before imatinib in chronic myelogenous leukemia. So I always think of this as kind of the low point in lung cancer research, and I point out, you know, our leading journal in the United States is the New England Journal of Medicine, and this sort of miserable outcome was what was getting published in the New England Journal in 2002, about 20 years ago. Now, the other thing that was going on at about that same time is that they were beginning to try to target one of the receptors on the outside of cancer cells. So epidermal growth factor receptor is something that's present in your skin. It's present in the lung cancer cells, and it has an extracellular domain here that sticks out outside your cell. This TK is a tyrosine kinase domain, and one of the things that happens is that we have these ligands that circulate around in your body, and these ligands bind to this extracellular portion of the receptors, and it stimulates these tyrosine kinase domain and ends up phosphorylating this and other proteins, and it causes the cancer cells to grow. So because it was present on the majority of cancer cells, the drug companies were taking 2 different approaches to try to target this. One was to end up generating an antibody, cetuximab, that bound to the receptor and prevented the ligand binding. And the second was to take a molecule directed against the tyrosine kinase domain in the intracellular milieu. And we're still, these 2 molecules are still around. Cetuximab and its relatives are used in colon cancer. It doesn't have much activity in lung cancer. And this is the structure of gefitinib. It's not terribly important other than it's made of this particular ring. So at the time in 2000, the monoclonal antibodies were developed against that extracellular domain of the extra epidermal growth factor receptor. And the gefitinib, which was one of the tyrosine kinase inhibitors, was developed to inhibit the function of the epidermal growth factor receptor. Now, this is a very popular idea, and gefitinib was given to hundreds of thousands of people around the country. And at the time, some of the patients had very dramatic responses. And I'm going to show you one of the patients from our center. And one of the things we began observing when these hundreds of thousands of patients were treated was seeing these very dramatic responses that instead of lasting for a few months was lasting for a few years. So this was a woman with adenocarcinoma. She'd never smoked cigarettes, and she got treated with gefitinib and had an exceptional response. And one of the things that happened, and one of the good things, it wasn't terribly complicated. The medicine was directed against the intracellular portion of the epidermal growth factor receptor. So working with two of my colleagues, Matt Meyerson and Bill Sellers, we ended up taking these patients. All this stuff is cancer here on both sides, and all this black stuff shows that it's gone away. And as you can see, it went away for about 2.5 years, something we almost never see. And we ended up sequencing this, and this was one of the original 5 in our paper that we published that had a mutation in the epidermal growth factor receptor. And I'm going to talk a little bit more about that in a moment. The second thing is that when I worked at the National Cancer Institute, we started permanent cell lines, took the tumor outside of the patients, and we'd grow it, and we could grow it in about a third of the patients. And one of the things I did as a postdoctoral fellow was I looked up all the ones what their characteristics were. And so this 3255 was from a woman who didn't smoke, which is where we typically see the epidermal growth factor receptor mutations, which we didn't know at the time, and sequenced this. And this part in green right here shows, this curve shows the cells that are being inhibited to grow. And this is the concentration going from 1 nanomolar up to 1 micromolar. The amount that you can get into a person's body is right around in here. It's about 200 nanomolar. And as you can see, this is about 100-fold more sensitive than the other two cell lines and to this molecule, gefitinib. And as I mentioned before, it wasn't that complicated to sequence the receptor, and we ended up finding an L850R mutation. And one of the things that happened, we had publications on the same day. The group from the Massachusetts General Hospital, which was published in the New England Journal, and our group here published it in Science on the same day, and ended up finding that 13 of the 14 patients who had these dramatic responses had a mutation either in this spot, the L858 position, or here in the exon 19, where typically at least 4 amino acids are deleted. We've subsequently learned these activate the tyrosine kinase and ends up sending signals where it phosphorylates and makes the cancer grow. And the other good news was that this technology was licensed to LabCorp, and the test is licensed, and it generates millions of dollars over the course of several years. And I actually get some of that. Now, one of the things that happened and what I showed you in that State of the Union is one of the things we try to do is first compare it to what standard therapy is, and then secondly is to develop molecules that are more effective against this. So this is a study that was done in Japan, where the frequency of the EGFR mutations is much higher. They were able to get testing that was available by 2005 or 2006, and then they took patients who had prospectively identified them with mutations and gave half of them that gefitinib and half of them what then was that existing therapy that I showed you in that first curve. And this trial was stopped early because you saw a 70% improvement in outcome. And that is the patients who had the mutation and got gefitinib ended up having their tumors grow at about 11 months, and the standard therapy where they got chemotherapy was only about 5.4 months. So a big step forward. This eventually translated into the patients living for years instead of months. So the next thing that happened is that we began looking at ways to overcome the cancer's resistance to therapy. And one of the things that happened is that they defined the most common mechanism of why it quit working. And the reason it would quit working is that in this particular position, where a threonine is supposed to be, which is a small amino acid, a bulky methionine comes in, and then the gefitinib can't get in there. And so this WZ402, and this was one of our postdocs from China, it was Wei Zhao was his name, and it was a 402nd molecule that he looked at. So he ended up designing a very different structure, a pyrimidine-based structure, that would end up binding in that pocket, and it worked when it had this T790M mutation. Now this is a little hard to see, but one of the people that, so here in the here's a mouse model that's got this this resistance mutation, and when you treat these mice that have these specific ones by using this compound WZ402, you can end up seeing that when you give the drug, when you don't give the drug, all this white stuff gets worse, and here when you give the drug it gets better. And it's the same whether you have this exon 19 or the exon or the L850R mutation. So this was published in 2009. A number of different pharmaceutical companies developed compounds similar to this. And we began testing it in people after they quit, when it started to grow on gefitinib. And as you can see here, this is one of my patients who had gotten gefitinib for about a year. And this white stuff here is her lung cancer. And this is cancer cells growing in the fluid. And there's some other material going on the right. And when this was in 2013, so it only took about 4 years between when that acquired resistance mutation was discovered and when the drug became available for clinical trials. And here, the patient ended up having the clearing of this lesion here on the left, as well as clearing of the pleural effusion that ended up working for about a year and a half. So this paradigm of taking an effective drug, finding out what caused the resistance, developing a new drug that works in previously treated patients, and then move it up further into therapy. So this shows what happens. They confirm that indeed if you develop that acquired resistance mutation, the threonine to methionine, the T to M, that this works for about 10 months, whereas if they don't have it, it doesn't work very well. And this is the structure of osimertinib. And osimertinib was approved in the US for acquired resistance in 2015, and in March of 2017 it was approved in China and is now the standard agent. And when you go in and compare this to the older EGFR TKI, which is gefitinib or erlotinib, and compare it to osimertinib, you can see this works for about 9 months longer. And then when you take a look at the survival curves, the red one is the older ones, and the blue one is the new osimertinib drug. So this is a drug that's used around the world. The worldwide sales are now in excess of $5 billion US. So by doing this, not only do you find out better treatments, but it also makes it financially viable effort to be able to do this and to find out how one can move from an effective drug to a more effective drug over the course, and this all took place over the course of about the last 20 years. I'm going to give a second example of the ALK rearrangements. ALK stands for anaplastic lymphoma kinase. It was originally discovered to be rearranged in childhood lymphomas, which mercifully are very rare, and hence the name anaplastic lymphoma kinase, and despite the studies that we made in lung cancer. Now, one of the other ones on President Obama is that one of his statements that he's made on precision medicine is to enable healthcare providers to tailor treatment and prevention strategies to people's unique characteristics. And that is to study what it is in the cancer that you can exploit that's going to make it work more effectively and improve the therapeutic index. That is to give treatments that are more effective and have lesser side effects. This was in the White House. My daughter worked in the White House during his first term. And one of the things that happens is that in order to be able to do this, you need to have the testing. And I'm going to show you some information about the testing. So the testing needs to tailor the treatment strategies to the people's unique characteristics. And these are two pictures of lung cancers. They're meant to look nearly identical. The one to your left is one that has an EGFR mutation, and the one on the right has an ALK rearrangement. You would never know that without doing the testing. So you can't, there are people that are trying to use artificial intelligence and other techniques to be able to find it without doing mutation testing, but but at the current time, one needs to do the testing on this to be able to sort this out. So this was a study that was done by in Japan, and they ended up finding 5 of 75 non-small cell lung cancer patients ended up having this transcription factor, something that makes the transcription go higher, and they ended up discovering 5 of these ended up moving the tyrosine kinase behind a transcription factor, which turns on this ALK kinase and makes the cancer grow relatively rapidly. And this is one of my patients. This became available in the late 2000s, and this is 2011 when I saw when this was just becoming available through a trial. And this is the person's This is the person's tumor mass. Here on the right side, this white stuff. And she was a very unusual person in that this worked for 7 years. This mass is no longer present here in March of 18, but she's developed this new one. So as happened before with gefitinib, we end up seeing that the person, this does work, but eventually it quits working. And one of the things we learned is that the crizotinib ended up doing the same kind of test where you compared this to giving chemotherapy. And once again, these patients end up responding for about a median of 10 months, and here once again for about 6 months. There ends up not being a difference in survival, but almost all those patients cross over to the treatment. The other part I would point out here in these patients is that, and I'll show you in a minute, is about how long this works. So one of the things that happened, as I mentioned before, the paradigm of taking the existing drugs, finding out why it quits working, and then developing drugs to work in the relapse setting, and then move it up front. So my same lady here, and here in this one that occurred 7 years after she started in March of 18, she's been on alectinib, which is a second generation inhibitor. She's been on it another 5 years. So she's been alive now for 13 years. And she started out with brain, it had spread to her brain. She had very advanced cancer. And her sons were in junior high school. They were in 8th or 9th grade, 3 years from finishing high school. They both finished graduate school, and her son's getting married this year. So we've been able to see her go through those stages of life by using these targeted therapies that are more effective than what we had in the past. So this is what happens when you, as I showed you before, what happens when you use this alectinib compared to the crizotinib. Once again, this is active in previously treated patients, so it got moved up front in this randomized trial comparing alectinib to crizotinib. And here you can see the progression-free survival. This one really levels out, and when you go into here in the survival, the majority of people are alive at 5 years. So this is no longer the deadly disease, and this one had the same outcome as we've seen in the past. I'd point out this is published here by one of your investigators here from Hong Kong, Dr. Mok, and I'll be seeing him later on this trip. So last thing I want to cover is patient outcome. And how does this show up if we take a look at a population where you can do this testing and see what the outcomes are? So this is data from the United States, and when you've taken a look at the age-adjusted number of deaths, this starts out at about 53 in 2001, where I started my lecture, and it's been declining. A lot of this is driven by smoking cessation in the US, but also, I'm going to show you in a minute, it's also in part driven by more effective treatments. The other thing is that these other two curves are, one, if they have a cancer diagnosis and the death certificate says they died of lung cancer, and the other one is they have a diagnosis of lung cancer and the cause of death. But as you can see in all three of those, the numbers of patients dying from lung cancer is declining. And one of the things they point out here, if they take a look at this, if they take a look at what's happened between 2001 through 2015, they identify an inflection point here at 2013 where the first targeted agent was approved. So the decline here before 2013 was at about 1.9% per year, and it then dropped to 3.1% decline. And the same thing here if we take a look here in the mortality, you see the mortalities drop from 3.2% to 6.3%. You see something similar in women. Once again, when you go here from the mortality and you look at 2013, it goes from 2.3% decline to 5.9%. Well, this could all be related to incidence and mortality, but when we take a look, when we take a look at the survival, when we look at non-small cell lung cancer, you can see that in men, the 2-year survival in the US has gone from 26 to 35%. And for women, it's gone from 35% to 44%. So the women live longer than men, and this has been true for a long period of time. So you're seeing not only a difference of people with these specific abnormalities, but you're also seeing a difference in our populations, that not as many, number one is not as many people are getting it for the public health reasons, but number two is if you do get it, the outcome is getting better, and it's increasing with the ability to identify these targeted treatments and to give people these efficacy. Now this is President Biden, and this is a statement from his White House fact sheet that science brought us treatments that target specific mutations in many types of cancer, for instance, in certain types of lung cancer. And this is his comments on precision medicine. And one of the things I want to show you, and one of the things I hope here in Hong Kong and in China, that people are getting this testing. It doesn't do any good to have these fancy medicines if you don't identify this. And I want to show you an effort that took a look at the oncologists in the United States. And that is one of the organizations that takes a look at a large structured patient database is called Flatiron. So they took a look nationwide at a health record database, and they looked at 30,000 adults with advanced non-small cell lung cancer diagnosed between 2015 and 21 who got their first-line therapy within 90 days. And the ones that were approved, where there were drugs approved for this at the time, were PD-L1, EGFR, ALK, ROS1, KRAS, and BRAF. I've talked about EGFR and ALK. All of these were approved within this time period. So the question comes up is, are people actually getting the testing so they could potentially benefit from these therapeutic agents? And this to me was a little bit encouraging and a bit depressing. So this top line here is if anybody got any kind of testing. So it's during that period of time, and it's gone up from 2015 up here to 2021 from about 68% to 85%. When you take a look at individual ones, now these go up very, very slowly. The uptake of doing the testing in the United States only goes up a few percent for each of these, despite them during this time period having approval for these agents. KRAS was approved after this, so we didn't expect this to be, there's a good reason why this is low, but the others were approved, somewhere between 2013 and 2017. The one that is a little bit different here is this is PD-L1 testing, programmed death ligand. That's a predictive marker for immunotherapy, and there was for second line for during this period of time in 2016, it was approved for patients who had 1% or more PD-L1. So this one took off very fast, in part because it's a simple test. It's an immunohistochemical test instead of a genomic test. So the ability to do it is much simpler. You can do it on regular past specimens in most routine labs. So with that, I wanted to show you just those two examples of EGFR and ALK and what's happened over the last 20 years. The innovation part is that those cell lines that I showed you, 3255 that has the EGFR mutation, the National Institutes of Health has licensed those cell lines for companies for drug development. 3255 is a favorite, 3122 is an ALK rearranged, and that also generates multi-million dollars a year as they get licensed to drug companies. These cell lines that came from patients with these genomic changes where they're annotated about what we knew about what happened with the patients has been a big boon to the National Institutes of Health. And I also get a little bit of that as well, which is a nice incentive. So I didn't set out to set up things to be able to license them, but these are things that are important and people have been willing to pay and license these for scientific advancement. So thank you.